for the invite to be able to speak at this symposium today. Unfortunately, because of time differences, I'm not able to attend in person or to join you live. But what I'm going to do in this presentation is we talk a bit about ICH Q3D, the guideline, focusing obviously on elemental impurity in the risk assessment process that's used to assess whether or not they are a concern or not. This is a little bit of background, something that I'm sure you're already aware of, but as far as ICH Q3D is concerned, from a basic perspective, we need to think about elements from the perspective of their toxicity or otherwise. And we start off with class 1 metals. These are the 4 elements defined as class 1. This is arsenic, mercury, cadmium and lead, often referred to as the big 4. And they are defined based not only on their toxicity, but also in terms of their high natural abundance, or at least perceived high natural abundance. And we will see more later as regards whether or not that indeed is actually truly the case, as to whether or not they are naturally abundant. And so one of the other key things that I want to comment on is the fact that when we talk about this risk of the big 4, is that what it means in reality is that you always have to assess the potential risk that is associated with the main 4, or the big 4 as they're referred to as arsenic, mercury, cadmium and lead. But the guidance talks about them needing to be assessed. One of the common mistakes that's often made is that many take this as, and misinterpret this as meaning you have to test for arsenic, mercury, cadmium and lead. You don't. This is just that, as far as the guidance is concerned, and understandably so, irrespective of the route, your risk assessment always has to take those 4 elements into consideration. So I'm now going to go on and start to talk about the other classes of elements. So there are class 2 elements, and these are elements that are considered still to at least have some concern from a toxicity point of view. And as far as they're concerned, the class 2 elements are actually split into 2 further subclasses. So you have class 2A elements. These are the elements that are considered to be to present a risk because of their at least apparent high natural abundance. And this includes elements like cobalt, nickel and vanadium. And then we have the class 2B elements. Whilst these are still a concern from a toxicity point of view, it actually relates to elements that are generally considered to be of low natural abundance. So in terms of risk assessment, class 2B elements need only be considered when deliberately added to the manufacturing process, typically as a metal catalyst. And the kind of elements that are focused on here are silver, gold and the others that are listed here. Out of those, the most obvious ones are palladium and platinum. And the next thing, in terms of at least the very simple basics around the guidance itself, is that the guidance itself actually established limits for 24 elements in total, and each of these are then listed in terms of their class, their limits from an oral, parenteral and inhalation perspective. And indeed, one of the reasons I'm often asked is why are there only 24 elements? Well, the reality is because those are a mixture of things really. One is that these are the elements that are typically associated with or attributed to potentially be present in pharmaceuticals, either through their use deliberately as metal catalyst or something of that nature, or that they may be present as a result of their natural abundance. For elements outside of that, the second reason as well is that for elements outside of that, there's a real lack of actual safety data for any other elements outside the 24. So if you are, as part of your risk assessment, ever looking at elements that fall outside of the 24 that are listed here, then obtaining the safety data may be quite challenging. And then each of the elements, as I've already said, is classified, and also within the guidance, what it does, it relates what elements you need to risk assess based on the route of administration. And so there are the class 3 elements, and these are the elements that are only required to be part of the risk assessment if you're talking about parenteral use or inhalation use. If you're simply talking about a simple tablet where you're talking about oral solid dosage forms, then the elements you would always look for are cadmium, the 2A elements, cobalt, vanadium and nickel, and any 2B catalysts that are perhaps possibly used typically in the synthesis of the active. The next thing is to think about how do we actually evaluate the risk associated with elemental impurities anyway. And the guidance is very helpful in this respect. It actually follows the basic principles as you would expect of any kind of risk assessment. In terms of, you have in line with the principles of ICHQ9, you have an identify phase, an evaluate, and then to summarize the control. So for the identify phase, essentially what you're looking for are any potential sources of elemental impurities. And what those are and how to break them down, I'm going to go through those in some detail as I go through the rest of the presentation. And then ultimately, once you've collected data, whether it's actual measured data or data obtained, for instance, from the perspective of from a literature point of view or published data or something similar, then you evaluate that. And ultimately, as part of the evaluation phase, what you're looking to do is look at the information in terms of what elemental impurities may have present, what level may be present, and how does that compare to the limit that's defined within ICHQ3D. And then ultimately, based on that, you decide as regards what the risk is, actual or just theoretical, and based on that, ultimately what control strategy is required. And all of this is about ultimately ensuring that you meet the permissible daily exposure limits. And it's an iterative process, and so you may go through these stages more than once as part of this as you go through the different phases of development. The first stage is the identify phase, and within the guidance, it has within it this fishbone diagram, which is quite useful from the perspective of thinking about where the risk may sit. Now, a key point to make as regards this is that the ultimate risk assessment in terms of ICHQ3D is focused on the drug product. It's the drug product that needs to be compliant. But the guidance provides this fishbone diagram, and within it spells out the different potential sources as far as elemental impurities are concerned. The drug substance, excipients, the manufacturing equipment you use both for drug substance and drug product, the utilities such as the use of water, how you store either your drug product or your drug substance, and ultimately looking at culminating the whole risk assessment in terms of what elemental impurities do I see in my actual drug substance. So let's break that down a bit. Let's look at this in a bit more detail and put a little bit more information and context around each of the different elements of that risk assessment process. So let's start off with what in many ways is considered to be, and in reality likely is, the biggest single source of risk, which is the manufacture of the drug substance. So in respect to the drug substance, one of the first questions you ask is, where in the process should the assessment start? Manufacturing processes for some drug substances are long and complex. You may have a synthesis that covers something like 20 chemical steps if it's a complex molecule. In general, though, it's reasonable to actually instigate the risk assessment from either the registered starter material or perhaps even, certainly for a low dose API, maybe you only have to consider the late stages, i.e. 2-3 steps in the synthesis to the API. And in most of the cases, the risk is associated with catalysts, heterogeneous catalysts like palladium or charcoal, where these are used or added to the process for a deliberate purpose, but then they're actually physically removed from the point of view of they may be filtered off or removed in some other way. And one of the things to note in regards to this is that because in Europe there was a previous guidance that covered metal catalysts, a lot of attention as regards impurities, elemental impurities, has indeed focused on the API. As it will turn out, as I go through this, in many ways that's warranted. And then if you look at the drug substance in terms of all the different aspects that could contribute to this, one of the first ones and most obvious ones is metal catalysts. These are the most likely source of elemental impurities in the drug substance, and these are present because they're intentionally added. You use a palladium catalyst for instance, a hydrogenation or a reduction or something similar to that. And indeed, the advent in 2008 of the European guideline has meant that many organisations already have, certainly for historical products, a very, very good understanding of the risk associated with metal catalysts and will have undertaken testing of the levels, and maybe even understanding of the potential purging of elemental impurities arising from a catalyst in a typical process. Equipment is another potential factor, at least. The guidance states that the risk associated with manufacturing equipment is generally higher when related to the manufacture of the active, as opposed perhaps to the drug product. And this is because you use more aggressive conditions. You use reflux, you may use really chemically corrosive reagents, and these are used in the synthetic process, but not less necessarily in the drug product. And while that's generally true, and in theory these are risks, of course we do all our manufacture of pharmaceuticals under good manufacturing processes, GMP, and indeed running processes on suitable equipment simply makes no sense, either in terms of the fact you may, in this instance, leach out metals that end up in your product and therefore contaminate the product, or perhaps even more relevant is you can't continuously replace damaged equipment. And GMP will typically involve an assessment of equipment compatibility as part of accommodating that process. You check whether or not your equipment is compatible with the process. And indeed, there are often deliberately designed experiments that show that elemental impurities are not leaching from manufacturing equipment into either the solution, i.e. the process liquors, or ultimately into the drug substance. Other factors are things like processing aids, organic materials. These are not likely to contain significant levels of elemental impurities. Water we'll come back to in a little while, but if you're using high quality USB grade water, it's outside the scope. Why? I'll explain. Solvents. Very few solvents utilize metals, certainly deliberately in their manufacture, and even if they were to use metals, many of these solvents are distilled, and the metals are not volatile, so they would not contaminate the product. Containers, for instance, storage of the drug substance, there's little evidence of any kind of contamination associated with packaging, either in terms of any metals associated with the packaging already, but critically, it's a solid-to-solid interaction. You have your solid drug substance inside of a plastic bag, inside of a plastic drum. That's solid-to-solid contact. There's no mechanism for which elemental impurities may migrate across that packaging. Moving into actual other factors, and perhaps certainly reflecting on the fact that when ICH Q3D was developed, there was a very strongly held and generally held view that excipients could be a substantive source of elemental impurities. And they were considered by many as the most likely source of risk. And it depends, of course, within that what type of excipients. And there's almost, or at least a believed to have been, a sliding scale from synthesized excipients that do not use any kind of metal catalyst to those like lactose or gelatin derived from animals, those derived from plant materials like cellulose, all the way through, and ultimately the one that was considered to be the biggest risk was the mined excipients. And of course another risk factor is water, and water is generally used in the manufacture of both the drug substance and the drug product. In parenteral formulations in particular, used for instance in large volume parenterals, it's the major component in the final drug product, so of course it is a potential risk. But the key thing to bear in mind here is the risk is directly related to the quality of the water that you used. And as with processing equipment, good manufacturing practice, GMP, is an integral part of the overall management of this risk. And indeed, the guidance actually states, as part of a standard GMP procedure, water quality should be routinely monitored, and the purified water system and storage of the water should not reintroduce elemental impurities. And if that is the case, then water is not considered to be a substantive risk. And indeed, as far as this process is concerned, you have to, as a minimum standard, use drinking water, WHO, drinking standard water, standard quality for water that's used from a drinking point of view, and that it should be used only in stages up to the drug substance final intermediate. But if you isolate the drug substance and you're using water, you should use purified water. Similarly, in the manufacture of the drug product, that requires similar exacting standards. And indeed, In terms of water that's used for drug product manufacture, that may even be to an even higher standard. For instance, water for injection. And indeed, all of this was covered by by a USP stimuli article. And what does that say? Well, the article itself says, alert from the USP chapter within the National Formulary. It basically, chapter 1231, water for pharmaceutical purposes, makes this statement. Water that meets the US EPA, which is equivalent to the WHO, National Primary Drinking Water Regulations, or indeed the WHO Drinking Water Guidelines, that has been purified by conventional technologies to produce water, can comply with USP Chapter 232. And if you weren't aware, USP Chapter 232 is essentially the ICH Q3D guideline, just incorporated within the pharmacopoeia. What about manufacturing equipment for drug product? Well, I mean, in many ways, the risk is lower than it would be certainly within the actual drug substance manufacturing. The conditions are less chemically aggressive. You avoid the extremes of pH or high temperature or indeed highly reactive reagents. Drug product manufacture in the drug product may still, of course, involve the use of solvents or certainly liquid formulations. And really the only risk is in regards to physically aggressive conditions that could lead, for instance, to abrasion. And as far as that is concerned, that's talking about things like tabletware. Fundamentally, unlike the manufacturing of drug substance, there is no way or mechanism by which you can purge any elemental impurities. But it's provided that you apply GMP, good manufacturing practice, and you make sure that you monitor your equipment, you clean it appropriately, you, for instance, regularly and routinely change, for instance, the dyes used on a tablet punch, then there should be no issue. What about packaging? Is packaging an issue? Well, within the guidance itself, it includes a statement. The probability of the risk of an elemental impurity leaching into a solid dosage form from packaging is minimal and actually doesn't require further consideration in the risk assessment. And indeed, a lot of work was undertaken and published in 2015 at the time the guidance was being developed that looked at the risk within the packaging materials itself. Now, trace levels up to 100 parts per million of cadmium lead were seen in some plastics in polyvinyl chloride, but that was total digestion in concentrated acid like nitric acid or even hydrofluoric acid. When you look at the deliberate extraction, for instance, when you cut up a bag like a PVC bag and reflux it in a solvent for 24 hours, then the extraction levels were 1,000-fold lower. And we're talking here about leaching the actual, what level would come out during typical use. Not digestion, not extraction, but typical use. And so these levels are incredibly low and, impose little more than a theoretical risk. Once you've identified all the different sources, the next thing is to, look at, is there an actual risk? Collect the predicted and observed data. What is the level of a particular element within a particular material? And when you have that, you can ultimately do your risk assessment in the final drug product and determine, is there a risk or not? Is control required, or even do you need to reformulate to bring about reduction of a particular elemental impurity? What it doesn't mean as part of the evaluation is that you screen for every element. You can, of course, using analytical techniques like ICCP, look for all of the 24 elements, in fact even more, simultaneously. But that's not a risk assessment. Your risk assessment should certainly contain an evaluation of the risk at first principles level, looking at the equipment, good manufacturing practice, the container closure systems, especially for solid. What about the quality of your purified water? And in particular, can you use existing data? As far as analysis is concerned, this really should only be used as part of your risk assessment if you have no data and you need data to inform your evaluation of the overall risk. Excipients, though, as I've already said, as far as excipients are concerned, for many, including those working on the guidance, and that includes myself on the expert working group, there was this real concern that excipients, particularly mined excipients, would be a major source of elemental impurities. But this is the critical part of the whole talk around ICH Q3D, is what has been the actual reality. And the reality is very different. And it started off with this publication in 2015. This is where the FDA itself actually looked at the levels of elemental impurities within excipients. They studied over 200 samples of different types of excipients, plant, synthetic, animal, and of course the one of most concern, or at least perceived to be of most concern, the mined excipients. And what did they find? Well, even within mined excipients, they found little evidence of substantial levels of even arsenic, mercury, cadmium, lead. Where it could be detected, for instance within excipients like titanium dioxide, a mined excipient, then the actual levels were relatively low, 10 parts per million. And certainly when you consider how much titanium dioxide, or more precisely how little titanium dioxide is used in a formulated product, where it's typically only associated with the aqueous film coating, i.e. levels of up to but no higher than 3%, then it is not a significant risk factor. And also critical in this was they found that the actual variability was not significant. They saw and reported class 1 elements like cadmium in materials that are of mineral origin, like magnesium hydroxide and calcium carbonate. They exceed the option 1 limits that are described within the guidance, but option 1 relates to a dose of 10 grams a day. You don't take these at 10 grams a day. And what they ultimately concluded, it was highly unlikely that the levels were so significant that they would adversely impact on the overall quality of the drug product. Generally, metals were seen where they were expected to be found. Class 2A elements, after all being talked about as being abundant, were seen in some mined excipients. But they were in materials like ferric oxide or ferric carbonate. And these excipients, if indeed they are used at levels well below 1%. So is it a risk? Is it an issue? In reality, when you look at the risk in the overall drug product, no, it's not, and this is the critical thing. But that was only a snapshot in time. And from an industry point of view, myself and others had discussions at a meeting, a joint pharmaceutical analysis group in the UK in October 2013. We were all asking the same questions. Have I seen elemental impurities at a level of concern in any excipient? And the reality was we were all saying no. But at this point we decided what it would make sense to do would be to pool that data, to actually build up more consolidated knowledge, to build a database, share the data, and ultimately interpret it and ask the question, did it compare with what the FDA had seen? These are the companies that were ultimately involved in. There's a significant number of pharmaceutical companies collecting that data, and more latterly, in these same discussions, we also got data coming from excipient manufacturers. This was ultimately published. The purpose of this publication was to show how elemental impurities in that database I talked about could be used in assisting the execution of a drug product elemental impurity risk assessment, as of course required by the guidance. What this demonstrated was that in terms of the database, that if it was used in conjunction with other sources of information, then from that point of view, it's a valuable source of information to allow you to complete the drug product risk assessment. And that ultimately that collection of this data helps to reduce the burden of analytical testing. And so it's published this paper, and it looked at this from the point of view of the levels of elemental impurities within the excipients. And the critical question is, what did it find? Well, what it found was exactly the same as what the FDA did. Not on 200 samples, but on several thousand samples. And it showed no overall evidence of substantial levels of even the big four, arsenic, mercury, cadmium, and lead. Where detectable levels were seen, for instance in things like titanium dioxide, the levels were actually quite low, less than 10 parts per million. And we've already said we don't use titanium dioxide in large quantities within a tablet. What this showed was, even over a 3-year period, and in fact much, much longer than that now, we're now still at the stage now of feeling that the database has served its purpose. Data has been collected from 2013 to 2025. We've never found examples of even mined excipients with large levels of elemental impurities of concern. So this has been the huge, I suppose, critical piece ultimately in the understanding of the risk as far as Q3D is concerned in elemental impurities. And of course then there's the evaluate phase. What were my risks, or at least my potential risks? Are they actually risk? What is the actual levels that I might see? How does it compare to limits described with ICH-Q3D? And the final step is to evaluate this. And the next stage of the process involves the evaluation, taking into account any specific data that's been obtained. This could either be through analytical screening, supplier information, or of course it could be that database I've just been talking about. And when the guidance was rolled out, the implementation working group for Q3D produced training modules, and one of those included within it basically this scheme, or rather these four different points in this process to conclude your product risk assessment. And the questions that it asks are: the elemental impurities may exceed the PDE, in which case then you would have a significant challenge and problem. It may be close to the limit, or it could be 30% below, or we could see no elements at all. And in reality, in the vast majority of cases, what we found is that we are very, very rarely in this scenario. We've almost never here, and in 99.99% of all examples we're here. We're not finding a risk. Why? Because elemental impurities and excipients are not the problem it was believed that they would be. Now, of course, when you're talking about risk assessment, you have to formally document it. You want it to be comprehensive, describing what you did to risk assess the API and the drug substance, the API, the drug substance, and the drug product, and any contributors to it. And as long as you consider those, the data associated with them, you can make a conclusion as regards, is this or isn't this a problem as far as the guideline is concerned. And the benchmark against which this is all judged is 30% of the permissible daily exposure limit. But I've already said this. When this was looked at in reality, most organizations, well, nearly every organization on nearly every product found out that there was no substantive risk. You were here. There were only some very, very esoteric examples which are often very high dose types of products where there actually was any kind of risk whatsoever. And those are few and far between. The vast majority of drug substance, drug product related materials, whether you're talking about solid dosage forms, parenterals or inhalation, do not contain within them significant levels of elemental impurities that would be considered a risk. Ultimately, the final stage in this process is the control. What do I need to do to control my elemental impurities? There are several options. The guidance is quite flexible in this respect. Option one, the simplest, is to just say that the level of an elemental impurity must be below a particular limit based on the assumption you would take 10 grams. Well, most drugs do not take 10 grams. There are a few examples where you may exceed or come close to 10 grams, but that is typically for very large dose products. Option 2A is to basically establish a concentration limit, which takes into consideration the individual dose rather than it being 10 grams. That's very rarely used. The preferred option is option 2B. This uses the concentration limits as far as elements are concerned in individual components, and then summing those together and deciding whether or not they are indeed a significant risk. The final thing that we need to look at, or final option out of this, is option 3. This is where you test the drug product. But that's not a risk assessment. And from that point of view, it is an option that I would rarely recommend doing, because it is, as I've said, not a risk assessment. Option 2B is where you pool the data together. It's the total dose of the elements within the drug product that's the issue. Critically, the guidance makes it clear that if the risk assessment has determined that a particular element is not a potential impurity in a specific component, there's no need to test. You don't need to test down to parts per billion to find out if an element is actually truly not present within a particular component. If it's assumed to be 0, then you can record it as 0 within the risk assessment. And it's particularly useful, this option, for evaluating actual analytical data and aligning it to specifications. And as I say, option 3 is to test. But in reality, testing is not an actual risk assessment. And it's often used as part of your product development, but it's not an actual part of the risk assessment as such. You're testing in quality in many ways with it. Even if you are below the limit, even if you're below 30%, which is the point at which you have to consider routine testing, you still need to make sure that that risk assessment and the risk factors don't change. It needs to be aligned to the principles of overall quality systems, ICH Q10. It includes things like standard GMP and change control. If you make significant change to your process, is that actually indeed recorded? And make sure that you're against this risk assessment, that's audited. You want to make sure that specifications remain up to date. And as I said, critically within this is change control. Are they changing their supplier as regards a particular type of excipient? In reality, most companies will probably encapsulate all these different elements, but the one that's most often used is option 2B, and actually you can plug in your actual data into a calculator, like this example here. This is indeed a spreadsheet, an Excel spreadsheet provided by IPEC Americas. As long as you know what your levels of elemental impurities are and your dose and the percentage used in each formulation, then you can do these calculations. And indeed, an overall summary as regards Q3D is listed below. Initial concerns over the potential significant levels of metals and excipients, particularly mined excipients, was very high. In reality, the reality is that the risk is very low. For most dosage forms, risk assessments can even be performed based on existing data. That elemental impurity database for excipients, process compatibility for drug substance manufacturing equipment and regards also as well the drug product, good manufacturing procedures, and our understanding of which processes are acceptable for particular types of equipment and vessels. It's extremely well known, and we can leverage this in our risk assessment. And indeed, ultimately, Q3D has turned out to be the guidance that lists a risk assessment process for an exceptionally low risk, that in terms of ultimate control and testing for elemental impurities, it's very, very rare. And finally, just to finish off, just to make you aware of the fact that originally the guidance was written in the context of oral, parenteral, and inhalation routes of administration. We're also very much interested in what about where you're talking about, for instance, exposure to elemental impurities through, for instance, cutaneous exposure, i.e. rubbing a cream into your arm, some form of, maybe some form of antibiotic cream or something else, or something for dermatitis. And when it came to establishing those limits, it became apparent that the data wasn't good enough to set individual limits, certainly not for cutaneous exposure. So instead, an alternative approach was taken, and this was where the cutaneous exposure, permitted exposure dose, PDE, permitted daily exposure, is actually based on simply taking the parenteral daily exposure and multiplying it by a cutaneous adjustment factor. And for all elements other than thallium and arsenic, that's 10. For as far as arsenic is concerned, the adjustment factor is 2, and thallium has no adjustment factor at all. You just apply the limit as defined from the parenteral perspective. There are 2 elements that are considered to be sensitizers, nickel and chromium. And in those cases, not only did we take into consideration the dose, but we also take into the consideration the actual limit from the point of view of the concentration. And that comes to a limit of 35 micrograms per gram per day, if you're talking about the cream. And so from that point of view, these have been added to the guidance, but in terms of risk associated with this, how, for instance, have we seen significant risk associated with dermal products? No, it's the reality of this. And all of this is captured within that, that guidance. And in terms of the conclusions for that particular, there's additional elements. What is being said is There's a new appendix being developed that's for cutaneous and transcutaneous routes. It's important to bear in mind this only applies to intact skin. It doesn't apply to applying to mucous membranes or to your eyes or to other routes of administration, including subdermal. It establishes the PDEs for all elements in relation to their potential systemic toxicity. Specifically for nickel and cobalt, it relates to also the potential of sensitization and therefore the actual concentration limit as well. Although it's not on the slide, what I will say in conclusion is that we can do a very systematic way of assessing elemental impurities, as hopefully you've taken on board and as you've seen as I've talked about. But the critical thing is that since the guidance was first developed, we've learned what the true extent of the risk is, and particularly for the original concern around elemental impurities in excipients, particularly mined excipients, that concern has not turned out to be the reality. But with that, I'll stop at this point, and I hope this has been helpful. Sorry I can't be there in person, but I hope this has been helpful to you as you look to continue to do your risk assessments as far as elemental impurities are concerned. And with that, I'll say thank you.